Atoms are the smallest unit of matter. They define different elements and they can bond together to form molecules. Amino acids are a group of 20 modular molecules that can be assembled in different ways, kind of like Legos, to make proteins. Proteins are a type of biological polymer, which is a large molecule composed of repeating units, in this case, amino acids. Different combinations of these building blocks give proteins different shapes and functions similar to a job. Structural proteins are the most abundant. These include collagen, which is found in skin, bones, and other connective tissue that can make up to 25 to 35% of the protein content in mammals. Another protein is keratin, which makes up hair, feathers, and thorns. Enzymes are another class of protein that help to perform biochemical reactions. Some examples of these are pepsin or trypsin, which are used in digestion to break down proteins in our food into smaller bits. Lactase, which breaks down the sugar lactose found in milk into its component molecules, glucose and galactose. Lysozyme, which is found in egg whites, helps to destroy bacterial cell walls. And DNA polymerase, which helps synthesize new DNA when cells replicate. One of the many cool proteins we study comes from a carnivorous plant native to South Africa called Drosera capensis, or the Cape sundew. Similar to the Venice flytrap, which is a relative, the Cape sundew eats its insect prey without the need of any mechanical help, such as chewing. Instead, the plant wraps its leaves around the bug and then uses many different enzymes called proteases, similar to how we use pepsin and trypsin in our stomachs. Unlike us, though, the plant's stomach is on the leaves, which are still exposed to the air while it eats, which can take 1 to 2 weeks. So on top of it being really cool that a plant eats bugs to get its nutrients and learning more about how it does that, we also wanted to explore how it might fight off microbes like bacteria or fungi to keep them from stealing its meal. One thing we have found is that there is a neat protein found only in plants called the plant-specific insert. It is a smaller part of a bigger protein that has its own job. Interestingly, when it is cleaved, which means it has been separated or cut off from the larger protein, it appears to help fight against microbes. The structure of a protein can have a big impact on its job or function. So we have set out to learn more about the Druserin 1 plant-specific insert, or D1PSI for short. So far, we know that it is roughly 100 amino acids long and stable up to temperatures of 80 degrees Celsius or 176 degrees Fahrenheit, and a pH between 4 to 7, which means slightly acidic to neutral. For reference, vinegar has a pH of 2.5, and the inside of a tomato has a pH of about 4.5. Although each protein has a unique amino acid sequence, also called primary structure, they sometimes can have very similar structures or structural components, especially to closely related proteins. This molecular fingerprint can be used to predict what the protein looks like, especially if structures of similar proteins have already been solved experimentally. We use computer software to match or align segments of amino acids from the new protein to those of a known template that had been previously identified. Lucky for us, other plant-specific inserts, including one from a potato, were available to help. The 3D print you have in your kits is of the predicted structure of D1-PSI and is basically a representation of the protein magnified by 10 million times. If you think it looks like a roller coaster or a few different kinds of pasta, then world-renowned scientists agree. First, let's look at papain, a protease from the papaya fruit, to demonstrate each part and translate this into protein scientist lingo. The spiral or rotini parts are called alpha helices. The normal track, or thin spaghetti, is called loops. And the Y track, or fettuccine, are called beta strands. Together, these are called the secondary structure, and while the actual amino acids don't exactly look like this, these models can help us more easily visualize properties of the protein and tell us a lot about how it might interact with other molecules. What do you notice about the secondary structure of the PSI? Be sure to let us know what you see in the comments below. Finally, the tertiary structure is how it folds or is assembled in three-dimensional space. The PSI was particularly interesting because it was predicted to have an open and closed position or state. The exact function of D1-PSI is not entirely known. However, our lab has found that D1-PSI can stop the growth of the microbe yeast, while the closed conformation, your 3D print that is in this state, has yet to exhibit any activity. It is known that the open conformation can interact with membrane molecules as well as interact by itself. Two PSIs coming together to form what is called a dimer, preserving its membrane interacting activity. While we still have a lot to learn about this protein, we think it could be useful in the future as a non-toxic pesticide or antimicrobial. Each time a protein structure is solved, the data must be submitted to the Protein Data Bank, PDB. This is an amazing collection with contributions from scientists all over the world. To date, over 170,000 protein structures have been documented here, which is an incredible feat. But to put it in perspective, it is estimated that humans have anywhere from 80 to 400,000 proteins, and this database, while primarily human, contains proteins from many other organisms. So we have quite a long way to go and could use your help. Chimera is a software developed at UC San Francisco with funding from the National Institutes of Health that enables many types of protein visualization. By downloading this, you can open the data files from the Protein Data Bank and look at the structure of that protein. An article published in the Journal of Visualized Experiments in 2017, Beltram et al. Provide a step-by-step procedure to prepare a model structure for 3D printing by increasing the thickness of secondary structure elements and including the hydrogen bonds that are an essential part of the stability of alpha helices to maintain the twist, and for multiple beta strands to form bonds that create beta sheets. Once the values have been changed in Chimera, the file can be exported in a .stl or stereolithography format. This format is commonly used amongst 3D printing softwares as a precursor to the file that will include all of the slices and print parameters. 3D printing is also called additive manufacturing because rather than carving something out of a starting material, it sequentially deposits layers based on 2D images or slices to build the final form. The two most common commercially available 3D printers are extrusion versus stereolithography. At the Corona Public Library Makerspace, you have a Lotz-Bolt extrusion printer. This means that the printer uses a filament or plastic rod that is pulled into a heated nozzle where it is softened enough that it can be deposited. The publication and its accompanying video utilize this style of printer. In our lab, we have both an extrusion and stereolithography SLA printer. SLA printers use liquid resins that can be cured or hardened through the formation of new bonds with ultraviolet light, also known as UV light. To create the layers, the laser shines from under the resin like a projection or silhouette. We use the SLA printer, the Formlabs Form 3 with the draft resin, to print the proteins for the kit because it enabled high resolution prints 4 times faster than the other system. Once completed, we remove the prints from the build platform and rinse them in isopropyl alcohol to remove any extra liquid that may not have dripped off. Finally, we set up additional curing by heating it to 60 degrees Celsius or 140 degrees Fahrenheit. While UV light shines from all directions in a mirrored box. Once the prints were complete, we cleaned them, removing all of the support material that ensures overhangs don't sag or fall away during the print. We use 3D printing in our lab to help students visualize proteins, as well as to make parts for analytical instruments that we design to help study biomolecules. We hope that you will have fun printing your own proteins. Please comment below and tell us and let us know what you think about proteins or 3D printing and post questions. If you chose to make one, we would love to know which protein you chose and why. Be sure to upload pictures of your prints.